Welcome back here. So for all of you, your journey started here when we were here last August and the September before. So we've started the day today with Erika, which was here as a private presentation. So I'm just here to give you a few details, couple of minutes, also because we're waiting for Paola to come back. So I have maybe a couple of minutes. So during these 3 days, we're covering different sessions. We're in the middle of Ecophysiology and Ecotoxicology this morning. Then this afternoon, after lunch, we'll have Ecosystem and Biodiversity. Session 3, Space and Technology, the use and space of technology for marine understanding and conservation. Science and Economics, but still in Science and Society. And tomorrow afternoon, we will have at the same time, so here we are in Salle des Actes for the whole day. Tomorrow, we will be at the Belvedere, which is just outside the castle. When you enter, it's just there, another room here. Consultancy and Finance and Fisheries and Communities. It's for Blue Managers, with Blue Managers, but you're also welcome, of course. As you know, Blue Managers are mostly online, so these presentations will be mostly online, except for Alice, I think, who is going to be here, and as you may remember, or will remember. These presentations will be online, but the jury will be in the other room, just across this one, the Salon de Musique. Voilà. But we will reintroduce you to that after. So this section 1, Ecophysiology and Ecotoxicology. To be honest, initially, we haven't assumed that you will assume that the order on the PDF will be the order of the presentations. And as Erika was private, we plan to put Erika at the end. So we'll start all together. But together with this little mix-up and the help of the tramway in Nice, it was nice to start with you, Erika, like really calmly, alone in the room. So Erika, Alexandra, Kinones, Bonia, I know it's important for you, I pronounce all the names, talked about enhancing phycocyanin content in spirulina through dynamical Darwinian selection under oxidative stress, internship done at Villefranche. We'll go ahead so we'll come back to your order, as it's your order, Victoire. We'll talk about improvement of ferment tolerance in an industrial microalgal strain for nutritional applications, an internship done in Portugal. Florane Tondu is going to talk about investigating the role of microplastics, specifically polyethylene as a vector of brevetoxins, an internship done at Monaco. And Isis Almeida is going to talk about assessing interspecific variation of the skeletal and calcite from subantarctic echidoderms in the context of climate change, an internship done in Spain. We're lucky to have here a lot of people who assessed your work during for the manuscript, and who assessed your work here for the presentation. So for the jury members you've here, of course, you have recognized Paola Furlan, professor at Université Côte d'Azur IRCAN. Voilà. You ready? Nadir Laifet, PhD at Université Gadir ICN. Steve Como, researcher at Sorbonne Université Love. And Anne Pajot, senior postdoc at CEA in Cadarache. Voilà. Also, your reviewers, so the reviewers of your manuscript, I would like to thank them a lot for those here on site and those who are not here but have assessed your work. So thank you again to Steve, Paola, Nadir, and Anne, but also to Giovanna Cristina Varese, professor at Université of Torino, Stéphane Roberti, lecturer at Université de Liège, Yasmine Botin, professor at Université Côte d'Azur, Ecosys. So all of these people participated to the assessment of your manuscript. Okay, so for the continuation of the day, yes, so we will skip the break, except if someone has really to do something in between the three presentations, because we're already a little bit late. Then we have a lunch, and I would like to take this as an opportunity to thank again, a lot, the MARRES coordination team, who has done a lot of work to prepare everything. So bravo. Okay, all right. You will see during this week a new face, I think, also, because we're welcoming a new member in the MARRES coordination team. You will see that, you will hear its tone of voice is a bit different without a French accent. We told ourselves maybe it's time to have a real British accent in the team. So he's British, and Thomas will introduce himself a little bit later. Also, it's a lot of work in the coordination, because you may know that next week we are welcoming 150 students and colleagues from IMBRC at Sophia Antipolis. Obviously, the rooms here are too small. And so also, if you want to participate, to be here, to exchange with the students, don't hesitate to ask us during the week. There's always a possibility for you to attend part of the event. Okay. Victoire. Victoire, t'es d'abord. Allez, on va y aller maintenant. Also, I would like you to make clear that do not leave, of course, during your presentation if it's not obvious. And voilà. So, ta présentation, Victoire, juste pour être sûr qu'on est bon, c'est celle-ci. Et du coup, tu as le petit. C'est possible de prendre le. Encore mieux. Upgrade cette année. Alors, c'est. Ah voilà, écoute, je vais là. So for everyone, the little upgrade of the year. So this is the big button is to go next, and the button before is to go previous. You will see you have arrows on it, so you should make it. Do you have a timer?
Let me get my phone.
Yeah, don't forget to bring a timer with you if you want. Our last point also, guys, and especially for the jury, these presentations are live online, and we have a set of microphones. It might be difficult sometimes to hear the one person speaking, but it's surprising how a little whisper can be captured by the microphone easily at the same time. So during the presentations, if you can try to avoid commenting or anything, it can be surprising online after that. Yeah, last moment, yes, they can come. Hurry. But now. 5 seconds. They're coming or not? On va tester, voir si ça marchait.
Impec.
Okay, come on, take a seat. Voilà, the first one is nice. Go ahead, Victoire.
Hello, everyone. My name is Victoire, and I will be presenting the effects of salinity and temperature on growth performance and biochemical contents for potential industrial application of a Chlorella vulgaris strain. So the subject changed, as you may notice. So this is the layout of my presentation. So the world population is set to increase up to approximately 10 billion of people by 2050, which consequently will provoke an increased demand in food and especially in protein supply, while the world resources are being depleted. In 2021, animal-based food production represented approximately 57% of the greenhouse gas emissions, while the plant-based food production represented approximately 29% of the greenhouse gas emission, which is less, however, still impactful because of its use of arable land, which participates in deforestation. So there is a need to find a new alternative protein source or food sources. So why microalgae can be considered as an alternative feedstock and more? Well, because of its rich nutritional profile and its non-usage of arable lands, because microalgae to grow generally needs fresh water or seawater, sunlight, and CO2. So aside the alternative feedstock solution, microalgae biomass can be used in animal feed, aquaculture, biofertilizer, and most recently for biofuel production and wastewater treatments. Today, the main subject will be emphasized on the value-added products. So, to obtain the highest biomass productivity after the industrial production stage, the goal is to have the most optimal growth condition, and to control the optimal growth condition, it goes through the cultivation types or strain improvements. So we have 3 different cultivation types, such as the photoautotrophic one that uses lights and CO2 as sole carbon source, and the heterotrophic one that uses organic carbon as source in absence of light. So various studies and literature demonstrated that the heterotrophic cultivation types is the best one, is able to yield 3 times more biomass productivity than the photoautotrophic one. However, is more subject to risks of contamination and is the most costly. So then it goes through also the strain improvements to isolate the best performing strains. And there is treatment and several methods for that, such as the selective method, adaptive laboratory evolution, and the genetic engineering, as we commonly know to create GMOs, and the random mutagenesis, which uses radiation or chemical like EMS authorized for field application, notably in Europe. So the species of interest today is Chlorella vulgaris, which is a freshwater microalgae species that is able to grow and thrive under different growth conditions in extreme environments. This species is characterized by its phototolerance, thermotolerance, and halotolerance, which means that this species is able to grow under a high variation of light intensity, high variation of temperature, and high variation of salinity. So the bioactive compound of interest of these species are mostly proteins, lipids, pigments, polyunsaturated fatty acids, and vitamins. So over already 30 to 40 years, Chlorella vulgaris has already been commercialized as a nutritional supplement in human feed, and also in animal feed, cosmetics, and pharmaceutical industrial application. However, the most recent one that's Chlorella vulgaris has been taking interest into are the biofuel production and the wastewater treatments and the bioremediation, especially for aquaculture. So the thesis outline is the following one. The objectives were to determine the growth rates, the biomass productivity, and the biochemical contents of a novel Chlorella vulgaris strain under photoautotrophically cultivation type, under continuous illumination, and under different specific stress regimes. So the specific stress regimes are the different salinity concentration and different temperature. And also it was grown into closed photobioreactors to control better the environmental condition. So here you can see the hypothesis. So H1 is our hypothesis of interest, which will be validated or not. So to get into the material and methods, well, all manipulations were performed a flow laminar chamber after autoclaving and sterilize all the media and all material to ensure axenic condition and to prevent any risks of contamination. Then the growth trial were performed during 14 days until the reaching of the stationary phase. Then to collect the biomass, the culture were centrifuged and the supernatant removed. Then the biomass was lyophilized until further analysis. So the first growth trial and the second one were performed under triplicates. However, the third growth trial were performed for temperature, were performed under duplicates. So throughout these 14 days, the growth was assessed through optical density, thanks to a micropatch reader, which has a lot more functionalities than the spectrophotometer, but also does spectrophotometer, and cell concentration with cell counting with the Neubauer standard chamber, then the dry rates. However, to find the biomass productivity, the dry rate was taken only every 2 days, compared that optical density and cell concentration that were taken every day, to avoid a loss of more than a 20% loss of the initial culture volume. Then for the biochemical analysis, which help us to identify, quantify, and study the chemical substances and processes, the crude protein contents were obtained by multiplying the nitrogen contents by factors of 6.25. The lipids were extracted according to black light and Dyer method, which holds high quality lipids extraction through methanol and chloroform use. The pigments were extracted according to Lish and Toller and Wellburn method, where acetone breaks down the cell walls and membranes to release the pigments. Then the ashes content was calculated after the biomass being burnt into the muffle furnace. Then the carbohydrates, which correspond to fiber and sugar, was calculated by difference after obtaining all of these results. So about the results in discussion, we will start with the salinity growth trials, so the table 1 is a summary of the results of the growth assays that are displayed in Figure 6. And despite that the 20 ppt culture holds the highest growth rate and the 5 ppt culture holds the highest biomass productivity and highest cell concentration, well, after performing statistical analysis, no significant difference were founded. The p-values were all greater than 0.05, so therefore the H hypothesis 1 was rejected. So moving on to the protein contents, it's suggested from the Figure 7 that the higher the salinity was, the higher the protein content is. However, since statistical analysis showed that no significant difference was found because all the p-values were greater than 0.05, H1 was rejected. Now, moving on to the lipid contents, we can see that the culture unexposed to saline concentration are almost two times bigger. The lipid content was 2 times higher than the culture grown under different saline concentration. And statistical analysis showed significant differences. Now, it was the same for the pigment contents, however, with a few differences. So the 10 ppt and 5 ppt, which are lower salinity concentration, yielded a high amount of pigments. So what to retain about the salinity growth trial is that salinity did not affected our strain because of the statistical analysis. And the literature said that Chlorella vulgaris threshold is known to be 60 ppt, which is 2 times higher than what we tested, which was 30 ppt. And literature also stipulated that some strains of Chlorella vulgaris might be euryalin, which means that it's resistant to different saline concentration. Now, moving on to the lipids and pigments, media with higher saline concentration negatively impacted the lipid productivity. And the literature explained us that the reactive oxygen species induces oxidative stress. Therefore, and they play a role in the expression of pathways controlling lipid biosynthesis. Then moving on to the pigments, media with lower saline concentration enhance chlorophyll A plus B and carotenoid content. So the literature, it was explained by the fact that chlorophyll is the primary target of salt toxicity, therefore reducing photosynthesis and growth. However, that means that our culture under 5 ppt and 10 ppt didn't reach the toxicity level. So now moving on to the temperature growth trial, all statistical analysis validated the hypothesis, which means that temperature affected the growth performance, the protein content, the lipid content, and the pigment content. So what's to retain about the temperature growth trial? Well, the culture grown under 28 degrees at 1 ppt was the best growth performance. Here, if we come back, you can see in green. So there were significant differences. And the lowest culture was at 35 degrees at 1 ppt of salinity. Then for the protein contents, the higher the temperature is, the lower the protein content gets. So this was explained by the rising of the temperature, which reduces protein synthesis. In another matter, for the lipids, the higher temperature at 35 degrees enhanced lipid productivity. And in this case, we've found no literature supporting our findings. And for the pigments, the best growth performance, which was under 28 degrees, was also for the pigments and was coupled with high salinity concentration, which enhanced the chlorophyll A plus B and carotenoid content. So this was explained because Chlorella vulgaris is known for producing more protective pigments, such as carotenoid, to reduce the oxidative damage. So, to conclude, different salinity and thermal regimes affected Chlorella vulgaris strain, growth performance, and biochemical profile. The reactive oxygen species seems to play a significant role in the response to environmental stressors. And for industrial application, high protein content in Chlorella vulgaris makes it valuable in aquaculture and animal feed, as well as in nutritional supplements for human feed. And the high chlorophyll content yield in Chlorella vulgaris is also of great interest in extracting valuable compounds like antioxidants for pharmaceuticals, industrial application, and colorants for cosmetic application. However, in this thesis, the emphasis must be put under the fact that the temperature growth trial were performed under duplicates compared to the salinity trials that were performed under triplicates. And also that the growth condition of the salinity growth trial might be not optimal. So for future perspective, we could start by analyzing the pigments, the astaxanthin, lupin, and violaxanthin contents by HPLC, because reactive oxygen species, while put under salinity stress, enhance the production of astaxanthin contents. And astaxanthin content is a high valuable compound for pharmaceutical industrial application. Also, it would be interesting to investigate into the bacterial role in Chlorella vulgaris thermal tolerance and halotolerance. Here is a picture showing a bacterial layer after centrifugation, which shows that it was a culture grown under 35 degrees, which it would be interesting to investigate into. And to continue, it would be interesting to study a broader range of salinity and temperature combination, and to conduct also scale-up trials in larger photobioreactors, because it's essential to validate the lab-scale findings. And to finish, random mutagenesis has shown over the recent years great promises into giving new mutants of microalgae strains that were able to give higher contents of lipids, protein, and pigments. And I thank you all for your listening.
Thank you, Victoire. So for the last presentation, you were not there, but Paola started the question, so maybe we can start with you. And then...
Okay. Thank you very much for your nice presentation. I have a lot of questions, but we want to ask them all. Maybe we can discuss after. First, on the last sentence you said about mutagenesis. Mutagenesis, you cannot use in foods, in aquaculture, etc., modified strains, only in cosmetics. And if the cosmetic product is from a modified strain, it cannot benefit from the Cosmo label. So is it really a perspective as
it has been, it's, well, some PhD students and a researcher, they did a study and they obtained high protein content profile of Chlorella vulgaris strain. And it's in the idea for the European Union to accept, to
allow more.
Yes, thank you, to allow the EMS as to authorize the use of EMS to obtain new mutants.
Yeah, good luck with that, because even with the natural, it's very difficult to be authorized. So, okay. Another question, if I may, about the pigments. You mentioned that with just acetone, you were able to break the Chlorella vulgaris cells and access the pigments. But I know that this strain is specifically hard to break. Did you check under the microscope if the cell wall was indeed broken or not? Because in some of the labs I worked in, we used the high pressure homogenizer to break the cells because it was super hard to break them with just the solvent.
So all the methods is not only acetone was used. There was like a mechanical and first, before the chemical disruption, there was mechanical disruption involved with the high with bead street.
Okay.
Yeah,
I get it.
Some material that helps to break down the cell walls. And also a comment that I can do about obtaining the pigments is thanks to the protocol, doing approximately 12 rounds of acetone to extract the pigments.
If you want to be more eco-friendly, you can use ethanol as well. But acetone is fine. It's really, you can have all the pigments with acetone. But in the long run, with ethanol, it works well.
Can come back to you for questions per remember it is a good practice.
Thank you.
Yeah, wait. I forgot to read a little bit. Yeah, you passed very quickly on your result on temperature, and I think there was some. You're also testing the interaction between temperature and salinity, right?
Yes.
So can you explain that a bit better? Did you see any interactive effect, any additive effect, or well, yeah, if you can just explain that a bit more. For I think it was for lipids. You had some interesting. Trends, right?
Exactly. So what happened is, after obtaining the results of salinity growth trial, we were interested into investigating more with the 20 ppt saline concentration, which is more high than 5 ppt and 10 ppt. So we were wondering what result could we obtain. So we can indeed see, so for the protein contents, there is no significant differences. I mean, between temperature there is, but not between salinities. However, like you noticed, for the lipid contents, we obtain a really high amount of higher amount of lipid contents when the culture was grown under 35 degrees at 1 ppt compared to the 20 ppt, which holded less lipid contents. And well, I didn't find any literature to support my findings, because in the literature, the temperature is supposed to reduce lipid contents. I mean, from the literature that I found, the lipid content was reduced due to higher temperature. And the most interesting one is for the pigment contents, that's really higher amount of chlorophyll A plus B content was yielded compared to other growth condition, because 30 degrees is known to be the best, the most optimal growth condition for Chlorella vulgaris, and coupled with 20 ppt, well, it gave enhanced chlorophyll A plus B and carotenoid contents.
Okay, thanks. I was also wondering, when you test your salinity, so at high salinity, I think you have like lower lipid concentration, and also the pigments are lower, right, at higher salinity. So how do you reconcile that with the fact that growth rate is not affected? You know, is there a link then between lipid, pigments, growth? How do you kind of combine those different physiological parameters to understand what is happening with your cell culture?
The thing is, the reactive oxygen. Wait. Okay, so there were no significant differences between the different growth conditions. However, you can still see that the 30 ppt culture was the lowest one here, which is explained by the salt toxicity, which being exceed, thank you, which is being exceeded, and with the culture of 5 and 10 ppt, well, it's the salt toxicity has not been reached. So, as a mechanical differences, the reactive oxygen species induce oxidative stress, which is involved into cell signaling, which enhance the production of protective bioactive compounds, such as the pigments.
Okay. Okay, thanks.
Yeah. Nadir?
On the perspectives that you wrote, what will be the main goal to use HPLC on the?
Because all of my pigments analysis were performed with the spectrophotometer, and the spectrophotometer only allow to determine chlorophyll A, chlorophyll B, and carotenoids. So the idea in my thesis work was to have more information about the pigments contents by using HPLC, which gives more information about it.
They are not visible in UV spectrophotometry. Or
we did not perform this analysis. It's
Okay.
as a future work in the continuation of what has been already done.
I don't know if you said why, but why this range of temperature from 20 to 35? Is it to simulate some?
So, well, coming back to the context, the world resources are being more and more depleted as we go forward, and fresh water included. So, no, sorry, this is about salinity. Because of climate change, it was interesting for us to study the effect of temperature to analyze what was happening with microalgae strain in order to obtain strain thermal tolerance because of the possible climate change and the possible cultivation of, for example, Chlorella vulgaris strain for biofuels, biomass, value-added products, or in the future in open ponds, which will be exposed directly to higher temperature.
Thank you, Florane.
I have several questions. First of all, I would like to go back to about the link between your temperature and salinity experiment, or even temperature only and salinity beside, on the lipids and on the growth. You say before that you have, if there is less salinity, you have more lipids. And if you increase the salinity and if you increase the temperature, you decrease the lipids. Do you have an idea? You say, okay, I have no information in the literature about this link between the lipids and the temperature and the salinity?
Subjected to temperature, like when we found it, higher lipids contents, right?
Yeah, at low salinity level. High lipid at low salinity. High lipids, and there is low salinity, independently on the temperature.
Sorry. We didn't find any studies to support our findings, and on the figures we can also observe that the culture grown under 20 degrees is higher than the culture grown under 28 degrees, and then when the temperature gets higher,
It's the same.
the lipid contents is being enhanced, and the
Lipid content is enhanced.
sorry.
Where did you see an increase in lipids here? At 35 degrees only. At 35 degrees, you have an increase of lipids.
Yes, but
My question is how you can interpret this decrease of lipids when you increase salinity, whatever the temperature is.
because it's the salt, the species is intoxicated by the level of salt, which we can correlate with this results when we find that the culture unexposed to salinities shows higher lipid content.
Okay, I saw that. So if there is a toxicity inside the cell, what do you expect on growth, on the amount of cells that are produced, if there is this toxicity inside?
We expect a negative impact on growth.
Do you see this negative impact on growth?
Yes, 35. The culture under 35 degrees.
Beyond the salinity.
So the one with at 20 ppt also was also lower, significant lower growth performance than the other culture.
20 degree, 20 ppt, there is no difference in growth respect to 20 degree, 1 ppt. There is no differences.
Yes, but also we have to take in account that this temperature growth trial was performed under duplicates, so therefore we might not have all the good.
Okay, so you expect that in reality you should have a decrease in growth because there is this toxicity of cells, of salts, of
lipid
salinity.
Yeah.
So hypothesis, yes. Okay, and did you look at your cells at the experiment? Did you have an idea if they are in a good shape or not in a good shape, or?
Yes, during cell counting, I was observing the bigger cells, so leading to an osmotic shock induced by higher salinities. Not osmotic shock, but
If you have high salinity,
osmotic pressure, sorry.
High salinity outside, you should have more shrinkage, that an increase of volume now.
Well, the trend that was observed with this chlorella vulgaris strain was to the cells were bigger than cultured under higher salinity concentration. The cells were much bigger than at day 5, because at day 14 they seem to adapted to the osmotic pressure.
Okay. Do you know if there is some techniques to see the shape, if the cells are in good shape or not? Do you have an idea?
Yes.
Use?
Thanks to the microplate reader here, there is functionality that helps us analyze the fluorescence of the cells, which indicates if the cells is dead or alive.
The autofluorescence. The autofluorescence of the cells can tell you if the cells, or you had to put a dye.
To put a what, sorry?
Put a fluorescence probe. Do you need to put something inside the cells to be sure that it's dye or not, or it's the autofluorescence of the cells?
No, it's the autofluorescence.
Autofluorescence.
Well, I think we're good. And if you want to ask a last question. Don't feel obliged.
No, I have a lot.
You have a lot? Okay, a very last question for you.
Very last, on the methodology, on the dry weight methodology. So you have different salinities than the ammonium format that you used.
Yes.
How are you sure it didn't break the cells and it didn't lose some of the cells during the process? And then,
Well, I'm not sure. It was the protocol that was given to me, and but what I can say is for the culture not exposed to saline concentration, it was the distilled water was used to wash the cellular debris.
okay, thank you.
Thank you, Victoire. I think we're good. Can go ahead with you, Florane. Good for you. Okay. Florane, no stress, you're good?
Yes. As a presentation.
So we'll just give a minute for everyone to sit down. Hello. And so, Florane, you have the next 15 minutes to present your work. Go ahead.
Okay, perfect. So hello everyone, so I'm Florane Tondu, so I will present my master thesis project based on investigating the role of microplastics, specifically polyethylene as vector for brevet toxins. So this project I will present you today is a completely new project that has never been done before. So to start by a small introduction of what is the main problem. The main problem is the proliferation of harmful algal blooms. So they are produced by algae that generate toxin and can represent a risk to the marine environment, to the human health, but also can be linked to the fish mortality. The consumption of contaminated fish or shellfish, but also the inhalation of aerosol, but also the direct contact with harmful algal bloom can lead to various health issues. For example, you can have a respiratory problem or skin irritation. The second main problem is the presence of plastics in the marine environment. So since 2021, 390.7 million metric tons of plastics are produced. So in this project, we will focus on microplastic, which have a size equal or less than 5 millimeters. When they are in the marine environment, microplastic can interact with the microorganism present. So these microorganisms will form a biofilm around this microplastic, and this process is called biofouling. The microplastic have emerged as a potential substrate for harmful algae colonization and transportation. For this project, we will study brevetoxin. So Carina brevis are dinoflagellates responsible of reptiles in the Gulf of Mexico and in the Caribbean Sea. They produce brevetoxin, which are marine lipophilic biotoxin, present in the colon water and dispersed in the benthic and pelagic biota. The brevetoxin concentration in the marine environment range to 1 to 14 micrograms per liter of brevetoxin, and we know there are different kinds of brevetoxin. But in this project, we will focus on one type. We will focus on brevetoxin 3. These brevetoxin are present in marine aerosols. These biotoxins have the particularity to be responsible of neurotic shellfish poisoning. And the different symptoms depend on the concentration of ingested or inhalated brevetoxin. So this is a short example of the kind of symptom you can have. So you can have excessive salivation, diarrhea, abdominal pain, or nausea. This toxin also acts on cell voltage gate sodium channels. For this project, the microplastic we will study are polyethylene. But the question is why? Why? Because they are used in various fields. They are used in transportation, in construction, but also in plastic packaging. And they represent 30.6% of the plastic polymer present in the ocean, so we can find them in the water column, but also in the sediments. For this project, we will study particles which have a size ranging to 500 micrometer to 600 micrometer. So for this project, we have 3 different objectives. The first one is to determine the vector role of microplastics. The second is to determine the adsorption and desorption time of brevetoxin by these microplastics. And the last one is to determine if the presence of biofilm will increase the adsorption of brevetoxin by these microplastics. So to respond to this objective, we have 2 main hypotheses, but you will see for each experiment, we realize we have an hypothesis for each experiment. So the 2 main hypotheses are the presence of biofilm will increase the adsorption of brevetoxin in the plastics. And the second one, in the presence of biofilm, the desorption will be faster than in absence of biofilm. So to respond to this hypothesis and objective, we have 4 different experiments. The first one is the most easiest one. We just take it's a biofilm formation, so we take a bottle of 2 liters of seawater, we put the microplastic inside, we change every week the seawater, we add nutrients to boost the biofilm formation, and we just wait. The second experiment is the adsorption of crystal violet by these microplastics. So this technique is used to determine the presence or the absence of biofilm around the microplastic present in the bottle. So for this experiment, the hypothesis is higher absorbance for microplastic with biofilm compared to virgin microplastics. I will not go deeper in this experiment. I will just tell you during our experiment, we confirm the presence of biofilm around our microplastics. But before talking about the 2 last experiments, I want to mention why we use radiolabeled brevetoxins. So we use tritium-labeled brevetoxin to measure the adsorption and desorption of brevetoxin present in the microplastics. So tritium is a beta particle which is fixed on the side chain of brevetoxin. We don't know where. We know the tritium is fixed, but no where we don't know. When we use scintillation liquid for our experiments, the beta particle will interact and produce light. And the beta count will measure the number of emitted light by the beta particle and scintillation liquid. And this emitted light measure will help us to know the concentration of absorbed brevetoxin and dissolved brevetoxin in the microplastics. We are not able to do this experiment with natural brevetoxin because we are not able to sow the brevetoxin in the seawater. So at this moment, we don't know what is the concentration of absorbed and dissolved brevetoxin. That's why we need to use radioactivity to mark the brevetoxin and to be able to measure this adsorption and desorption. So I talked about adsorption. So the next experiment is adsorption of brevetoxin by the microplastics. So during 24 hours, we contaminate whole macroplastic with radiolabeled brevetoxin, and we measure three conditions: the virgin polyethylene no rinse, the virgin polyethylene rinse, and the polyethylene with biofilm. For this experiment, the hypothesis is higher brevetoxin adsorption in presence of biofilm. And the last one is the desorption experiment. So we measure the desorption after 24 hours of contamination of our microplastic, and we measure 1 hour, 6 hours, 24 hours, 72 hours, and 1 week of desorption, the concentration of brevetoxin present in the microplastic, but only in two conditions, for virgin polyethylene and polyethylene with biofilm. So for this experiment, the hypothesis is a faster desorption in presence of biofilm. Concerning the results for the biofilm formation, after 1 month, we are able to observe the formation of a biofilm around the wall of the bottle. And the presence of biofilm was confirmed around the microplastics thanks to the crystal violet adsorption by microplastics. Concerning the adsorption experiment, so we will start by virgin polyethylene no rinse. So what we can able to see, we are able to polyethylene no rinse absorb more brevetoxin compared to two other conditions. They absorb 28 nanomole per milliliter of brevetoxin. Compared to the virgin polyethylene rinse that only absorb 13 nanomole per milliliter of brevetoxin. That means rinsing the polyethylene, the virgin polyethylene, have an impact on the quantity of brevetoxin they are able to absorb. If we have a look on the polyethylene with biofilm, we can see they are able to absorb only a very small concentration of brevetoxin, only 2 nanomole per milliliter of brevetoxin, which is very, very small compared to the two other conditions. We can observe also we have a statistical differences between the three conditions concerning the brevetoxin adsorption. Concerning the desorption experiment, so for the virgin polyethylene, we realize a kinetics of desorption. So what we can observe along the desorption time, we have an homogeneous decrease of the brevetoxin absorbed by the microplastics. We are able to measure the half desorption time, so T1 and 2 per second. So that means the time need to desorb 50% of the brevetoxin absorbed by the microplastics. For this experiment is 80 hours. So we need 80 hours to desorb 50% of the brevetoxin absorbed in this experiment. We have an R-square at 0.87, which is close to 1 and which can represent what we have in the reality. But if we have a look after 1 week of desorption, we still have the presence of 25% of the brevetoxin activity in our microplastics. So that means we don't have a complete desorption of these microplastics after 1 week of desorption. Concerning the desorption experiment kinetics for polyethylene with biofilm, we observe we have a complete different kinetics of desorption. That means here we have a kinetics in 2 steps. The first step, since the beginning of the desorption to 6 hours, where we have a very fast desorption of our microplastic. You don't know, but here the desorption time is 0.49 hours. So we need only 0.49 hours to desorb 50% of the brevetoxin absorbed in this first part of the kinetic. The second part is very longer. We have a very small desorption of the microplastic. And this is confirmed by the half desorption time, where we need 160 hours to desorb 50% of the brevetoxin absorbed, which is very long compared to the virgin polyethylene and compared to the first part of the desorption. But again, after 1 week of desorption, we still have the presence of brevetoxin activity in the microplastics. So that means we still doesn't have a complete desorption of the microplastics. So now concerning the discussion about the adsorption experiment, so the virgin polyethylene rings absorb more brevetoxin compared to the virgin polyethylene rings. So we have an impact about rinsing the polyethylene concerning the adsorption of brevetoxin. The polyethylene with biofilm are able to absorb brevetoxin, but only a very small concentration. This result, so our test suggests that brevetoxin have a higher affinity for inorganic surface compared to organic surface. We know in literature, the biofilm play a role in the adsorption of heavy metal in the microplastics, but we are not able to do any comparison. For example, Penenal in 2023 show the biofilm help to increase the adsorption of heavy metal in microplastics, but this is not what we are able to observe in our experiments. Concerning the desorption experiment, so the brevetoxin desorption is observed in both conditions, so for virgin polyethylene and polyethylene with biofilm. But after 1 week of desorption, we still have the presence of brevetoxin in our microplastics. As I said before, the biofilm play a role in the adsorption of heavy metal, but also it plays a role in the desorption of heavy metal by the microplastics. But we are not able to do any direct comparison about what we do in this experiment and what has been done. Why? Because we study organic molecule, and we know organic molecule doesn't have the same behavior compared to inorganic one. So that's why we are not able to do any direct comparison about what we do in this experiment and what has been done before. Concerning the perspective, so to study the behavior of brevetoxin and the interaction between the microplastic, the biofilm, and the brevetoxin, we can do a lot of experiments because this is the first experiment to study this interaction. So the first one can be extend the duration of the desorption experiment to 10 days, 15 days, or 26 days. The goal can be to see after how many days of desorption we have a complete desorption of our microplastic. If we have one, we don't know. The second can be the DNA analysis to identify the biofilm composition. We know in the marine environment, we have a lot of microorganisms. We have a lot of bacteria. Maybe this composition, this bacteria composition of biofilm can have an impact on the brevetoxin adsorption or desorption in the microplastic. So that can be interesting to determine the biofilm composition, and if we change this biofilm composition, did we have the same result or not? The third test can be tested with saxitoxin. So saxitoxin are biotoxin produced by algae responsible of pelagic shellfish poisoning and also present during reptiles. But this molecule are smaller compared to brevetoxin. So that can be interesting to see when they're in contact of microplastic and biofilm. Saxitoxin, did they have the same behavior than brevetoxin? Did we have the same result or not? So we need, I think, to realize this experiment to compare the behavior of different marine biotoxins. The last test can be tested on marine organisms, for example, sea cucumber to prevent the seafood safety risk. So to conclude on this project, so this project is a new approach to study the interaction between microplastics, biofilm, and brevetoxin that has never been developed and explored before. The treatment label brevetoxin allows us to better understand the role of microplastic as carrier of marine biotoxin. We observed during this whole experiment, the biofilm limits the adsorption of brevetoxin by microplastics, but the desorption of brevetoxin is possible in both conditions. But we need to make additional experiments to better understand this interaction between the microplastics, the biofilm, and the marine biotoxin in general. So thank you for your attention. I hope this presentation was interesting for you. If you have any questions, don't hesitate. But first, I want to thank all the IAEA team members, Carlos, Marc, Nathalie, François, Khalil, and Emeline, for all their advice, for their scientific counsel, and also for their support in the creation and in the realization of this project. So thank you.
Thank you, Florane. So let's go for the question, starting with you. Yes.
Okay. So at the end, when you, for your perspective, when you said the other studies, you didn't mention, like, environmental drivers. And so you don't think that pH temperature could change even the toxicity of the toxin itself? Do you know if there is something on that, or?
And at this moment, we don't think about the physical parameter we have in the water, because we have physical parameters that can change for the microplastic also and for the toxin. And we use direct toxins that radiolabel, so we don't produce by ourselves the toxin. It just we buy the radiolabel toxin to the lab and we use them directly.
And so since you're talking about the radiolabel, do you think there is a risk that the tritium will attach to the plastic and not to the toxin at some point, that they will kind of move from one to the other, and that then what you will measure, what's left over in the plastic, is actually not the toxin, but just the tritium attached to the plastic?
About what we know at this moment, the tritium is directly attached to the molecule, and we are not able to dissociate the tritium and the brevetoxin. So normally, we have a good absorption of the brevetoxin labeled by the tritium, and not only the tritium on the plastics.
Okay. Yeah, I have another question. We say try to do two per person.
I mean, they are short, so you can ask a third one if you want.
Yeah, but I need to read my notes. My handwriting is really bad. So, yeah, I guess, what is your hypothesis to explain the fact that the toxin were not really attached to the plastic when you had this biofilm on them?
We think, yes, personally, I think it's because the brevetoxin acts on the sodium gate voltage channel of the cells, and bacteria have also this kind of channel on the cell. So maybe the brevetoxin acts directly on the biofilm, and that's why we are not able to be absorbed by the microplastic. But it's just an hypothesis, and we want to study that, but we don't know at this moment how we can measure that, how we can define that. Maybe we are thinking to develop a biofilm on the plate and just add a brevetoxin and see by the microscope if we have some cellular modification or not. This is hypothesis. We don't know how to manage that at this moment. So we need to discuss more with my supervisor how we can do that. But we can do really a lot of experiments because nothing has been done before. So 1 year, I will say for my internship, 1 year, and 1 year is too short to study everything. So we need to make choice.
Thank you, Steve. So before going to Nadia, just to recontextualize, to make sure, so you are in the middle of a 1 year internship and it's only a milestone after 6 months.
Exactly.
Nadia, then Paola.
Okay, so you used polyethylene.
Yes.
And I was wondering if you could, what other plastic would you want to try?
We can try with all type of plastics. We choose polyethylene because it's one of the most present in the marine environment, but also we have high polyethylene density, we have PVC. We can try it with all polyethylene particles, but we choose plastic particles. We choose polyethylene because we have them in the lab, and also we know with the size of the polyethylene to study the test on marine organisms. The animals are able to eat this size of microplastics. But we can do this experiment with all type of plastic and all type of marine biotoxins.
Because polyethylene is, that there's no oxygen on the chains. And for instance, if you try with PET, maybe it can be interesting to see the results also because you have oxygen, etc.
Yes.
And for the kinetics, do you have any idea of why this one? Yes. Do you have any idea of why you have this behavior?
No. This is a problem. We try to measure the presence and absence of biofilm along all the desorption experiments. I will show you. I have some results at the end. No, I don't have it. No. Yes. So we measure the presence of biofilm for the virgin polyethylene and polyethylene with biofilm. At the beginning, we are thinking during the desorption experiment, the biofilm removed from the plastic, but we know it's not possible because it's really attached. But maybe we can have a modification in the biofilm condition, biofilm composition. So that can explain why we have this fast desorption and after more longer. Maybe we have a modification, so the brevetoxin are absorbed by the plastic. And when we have this modification of composition, the brevetoxin are blocked inside the plastic particle. So that's why what we can think for this experiment, but I didn't tell you if we have statistical differences or not. I didn't have the time to do that, but it's a possibility. That's why we need to make DNA analysis to determine the biofilm composition.
Nadia, Paola?
I still have a question on that topics. And I would like to, could you explain me how the crystal violet will work?
Yes. So for the absorption of crystal violet, so we just take our microplastics into water, we put crystal violet, and we know crystal violet will interact with the cellular structure of the biofilm. So the cell which forms the biofilm will absorb the crystal violet, and when we will use ethanol at this step, we had ethanol to remove the biofilm attached to the cell, we will have an absorbance of crystal violet. And this is the crystal violet only present in the biofilm.
You said before that perhaps your biotoxin can have an impact directly on the cells, a toxic impact directly on the biofilm. Do you think that with this method you can assess this or not?
I don't think so, because this method is only a qualitative method to inform if we have biofilm or not. So maybe if the...
Not quantitative?
It's not quantitative. We don't have information about the quantity of biofilm formed around our plastics, because it's a very long experiment, but we need to do this experiment because if the quantity of biofilm change, maybe the absorption of brevetoxin change too also. So...
Okay, so I changed my question. Is there a way to assess the quantity, the quality in terms of viability of the cells of your biofilm in the presence of the biotoxins?
At this moment, I didn't tell you yes or not because I don't know. We need to go further in the project to be able to determine if we really have an impact or not. At this moment, I'm not able to tell you yes, no, maybe.
Maybe. A last question, a short one.
Yes.
I read your manuscript. I have the pleasure to read your manuscript. And in the manuscript, I understand that you change, you have two different amount of plastic in your two conditions. In order to have the same weight, you compensate by the same number of particles.
Exactly.
But as we are speaking about absorption, do you think that to increase the number of particles can have an impact on the amount of biotoxin that could stick on the plastics?
No, because when we increase the amount of particles, we also we don't decrease. I don't remember. No, normally no. Normally that doesn't have any impact because yes, we double the activity when we spike the brevetoxins. To be sure, we put 2 times the concentration of brevetoxin we put at the beginning of the experiment for the desorption only. Because for the desorption, we need
absorption only, but for the absorption one, because you find it's very interesting that with the biofilm you have less absorption than without.
Yes.
But you put also less particles when you do the experiment with the biofilm.
Yes.
So just thinking about it, there is not a bias due to the amount of surface that it's available for the biotoxin.
Yes. Normally that doesn't any impact. Normally.
Why you say that?
Because we count to have the same number of particles and they are in the big vial, so the surface available is the same for the both conditions. We double the quantity of biofilm with brevetoxin because we count the number of particles with brevetoxin to have the same weight. And to have the same number of particles to be the more precise possible, we need to increase the number of polyethylene with biofilm because the biofilm weighs a lot. So we don't really focus on the weight, we really focus on the number of particles for this experiment.
I understand, but I think that perhaps it could explain your different results. You have to test it, I think so.
Maybe, yes.
Thank you, Paola. You don't have a lot of questions?
No.
No, really?
No, but I still have one.
Great.
Thank you for the very nice and dynamic presentation. We can tell that you really love your subject.
Yes.
My question was, the first one was, will you continue your internship? But the answer is yes. So I am glad for you.
Yes.
Yeah.
Continue. Yes.
And it's a general question on an upper perspective. What is the point about the harmful algal blooms? Do you plan to put microplastics wherever there is a microalgal bloom and then collect the microplastic to avoid the toxins to be disseminated?
We want to do that in lab because we are not to do this experiment in the marine environment because we have too many factors. We know when the microplastics are in the marine environment, we have a degradation of the microplastics. So it's too difficult to have the same number of particles, the same weight, the same shape of particles. So we can realize that in lab to make the harmful algal bloom situation with approximately the concentration of brevetoxin we can have during this event and reproduce that in lab with a microplastic where we have the same shape, the same weight, and the same everything.
Okay, but what is if there is one, the application of this work?
This application is to give us an idea of what we can have in the reality. If we are not able to really show what we have because of the plastic degradation and everything, we can have an idea of the plastic brevetoxin absorption by plastics and the brevetoxin desorption. It's to give you a preliminary study of what we could have, what we can have in the marine environment, without any very nice representation, very good representation of what we have. It's just preliminary study to think about, okay, if we have microplastic and harmful algal bloom in the same time, what we can have. What can be the adsorption, the desorption if we have. It's just to put in the main problem, major problem we have at this moment about harmful algal bloom and plastic. If we combine both, what we can have.
Okay, and a very last question, is the toxicity of the brevetoxin is inhibited while it is adsorbed on the plastics, or is it still active?
We don't know. That's why we need to test on marine organisms. Because when we test with marine organisms, we have to study if the microplastic have an impact on the marine organisms, the brevetoxin have an impact, and also the combination of two have an impact on the marine organisms, but also at different time, because during the bloom event, we saw we have a desorption. So before the desorption of microplastic, what can be the impact? After a long time of desorption, what can be the impact on marine organisms? So we can have a lot of experiments to do.
Okay, okay, thank you.
Do you have a last question? We have time for a last question.
I have a last.
Question.
Okay, here you use external biotoxin. Use biotoxin that it's free in the water.
Yes.
But this biotoxin at one point is produced by an algae, is produced by organisms. How do you think that these organisms can interact with your plastic? The organism that produce the biotoxin?
We don't know if the algae that produce biotoxin interact with the plastic at this moment.
There is no work on that?
No. This is really the beginning, I think, of the great work we can do between the interaction of plastics and marine biotoxin. At this moment, a lot of study the impact of pollutants, heavy metal on microplastic, but not on toxin and harmful algal bloom. So I don't know at this moment if there is an interaction between the algae that produce toxin or the microplastics.
But if the algae can stuck inside on the plastic, do you think that the biofilm could help or not to trap or to fix the algae? I don't know.
At the beginning,
I have no idea.
At the beginning of my project, I will tell you yes. But now I'm not sure because we saw the biofilm doesn't help to absorb brevetoxin. So maybe for the cellular aspect, I don't know. I think it depends on the available surface we will have on the microplastics. If they will have enough available surface with the biofilm, maybe the cell can be stored on the microplastics. But I don't know.
Okay. Thank you, Florane.
Yes, thank
Just before you upload, I just wanted to tell you also, Victoire, that I forgot to share that. Thank you also for those online who are showing their support. Thank you, Marie-Cruz, Lucia, Nicole, Margot, Ilaria, Cassandra, who are saying hello and congratulations. For example, you can see in front of you that you have some comments. So you can continue online to share your comments. Congratulations, Florane.
Thank you.
We're back. You're more than ready, I can see.
Yeah.
So here we go for the last presentation of this morning. With you, Isis, and to introduce, of course, your work. You have the next 15 minutes.
Perfect. Hello, everyone. My name is Isis Almeida, and today I'm here to present to you my thesis defense. I've been working in Barcelona at the Institut de Ciencias de Mar with Ms. Blanca Figueroa on the theme assessing inter- and intraspecific variations in the skeletal magnesium calcite from subantarctic and Antarctic echinoderm species in the context of global change. This presentation is going to go the regular route. To start out with our introduction, with a little bit of context on ocean acidification and marine calcifiers, we know that since the beginning of the Industrial Revolution, the concentrations of CO2 in the atmosphere have been increasing rapidly. Only partially of these concentrations are able to be retained in the atmosphere, and around 30% of it is absorbed by the oceans, causing a process that I know we're all familiar with, which is the ocean acidification. The ocean acidification is a chemical process that tends to lower the natural pH of the water, turning it more acidic. This leads to the dissolution of the calcium carbonate skeleton of marine calcifiers, such as the echinoderms, and also lower the carbonate ion and carbonate saturation state, which are vital factors for the biomineralization of new organisms. Although these calcium carbonate shells are usually made out of calcite or aragonite, but they are often substituted by magnesium. Although they are not really clear on the reasons for this substitution, there is a link between the presence of magnesium and the higher temperatures, which is known to also potentialize the vulnerability to dissolution. To go a bit about where we're going to study, the Southern Ocean was created 34 million years ago when the Antarctica and the America drifted apart from each other, creating the Drake Passage. It's one of the youngest ocean basins on the world, and it's a primary mixing spot between the biggest oceans. It has a great influence in the world's circulation and a local and global climate. So why do we study the Southern Ocean? The Southern Ocean is supposed to be one of the first affected places by global warming, since CO2 is more soluble in cold waters. A region that is bathed by these waters are the subantarctic islands, which are found in the adjacent part of the polar front and north of it. They are known to have large ice caps, but they have shown great retraction in the past 40 years. This could be explained by different oceanic and atmospheric conditions. Usually, the global warming promotes the melting of the glaciers, which inputs fresh water, demising the salinity of the seawater, affecting the benthic fauna. Another place that is also bathed by these waters is the Antarctic, which is the continent encircled by the Antarctic polar front, so in this part. This is one of the fastest warming places on Earth, with the western part being the most affected. This doesn't mean that the eastern part is not, that it's safe, because a few signs of climate change have been seen in the past few years, with even a record-striking heat wave in 2020. So the benthic fauna is very affected by these changes, and today we're going to focus on the echinoderms. The echinoderms are part of the phylum Echinodermata with 5 different classes. They are deuterostome animals, and they have key roles as carbon sinkers and as predators in the benthic ecosystem. Although we've seen that they are intrinsically vulnerable to climate change impacts, studies have shown that they may have developed physiological tolerance to fluctuating conditions such as the pH and the temperature. But there is a lack of information, and most of these species are still understudied, so we don't know exactly how the biotic and abiotic factors can influence the skeletal mineralogy of these species. So it's really important to study these species from the subantarctic and Antarctic regions from the Southern Ocean to not only protect them, but also better manage the areas that are supposed to be affected later, such as the ones in the lower latitudes. This comes to me for our objectives. The objective is to understand how echinoderms might respond to environmental changes by analyzing the skeletal magnesium calcite in different species found in Antarctic and subantarctic regions. Our specific objective was to generate new mineralogical data, assess inter- and intraspecific variability in the magnesium calcite, and investigate the potential influences of environmental and biological factors in the variability. Thus, we came up with this hypothesis. We hypothesized that there will be intraspecific, so between the different skeletal parts of the echinoderm, and interspecific, so between the different species, variations in the skeletal magnesium calcite levels according to the different regions and due to the environmental and biological conditions. To test this hypothesis, we're going to focus on the Kerguelen Islands, which is a part of the French territory in the subantarctic region, which is a remote location known for their jarring climates, also known as Desolation Islands. Very interesting name. And they have a very interesting biota to analyze because the area, as we saw, it's already vulnerable to climate change and have heavily impacted benthic ecosystem. They are also, you can see it here in the map, a little bit closer to is above the Antarctica. Another place we're going to be talking about is the Terre de l'Île, which is part of the French territory, and the George Villaine, which is part of the Australian territories, in the eastern part of the Antarctic region. So not the main one that it's now the worst part. They also have extreme low temperatures and blizzards, and this may have led to the development of endemic species. They have a lack of anthropogenic influence because it's a very remote place, which led to simpler physiological features. For example, the absence of shell-crushing predators led to the echinoderms forming thinner shells, which could exacerbate the effects of the ocean acidification on them. We're going to focus on 12 different echinoderm species from 2 different subclasses, the Eoechinoidea and the subclass Sidaroidea. And here, just a picture to demonstrate all of them. I can't talk about it. I'm going to talk about them later. For the sampling, the species were collected in 4 different campaigns along the years. For the Kerguelen Island, the Poker 2 off the coast of Kerguelen and Protected 9 near the coast of Kerguelen. In Terre de l'Île during the Revolta 4th campaign, and in Terre de l'Île and George Villaine, the Siamark retreat. We have a table at the end in the annex to see the relation between species location and the areas in the campaigns. The processing was led for the separation by location and taxonomy. For the regular species, we decided to excise the test, which is the body of the echinoderm, to retract all the organic matter, and collect the Aristotle lantern, which is the mouth apparatus of the species. And the spines were plucked out. For the irregular species, which is the Abatus genus and the Brisaster antarcticus, the same thing was done for tests, but there was no presence of Aristotle lantern. They are usually sedimentary feeders and they don't have this mouth apparatus. And the spines were scraped from the test with a dissection blade rather than plucked out. Then the samples were left to air dry for 36 hours in the lab. The mineralogical analysis was done from 1 to 5 replicates from each station. The test spines and lantern were powdered and then put in a diffractometer to do the XRD measurements that would give us the magnesium calcite values. Once we got these values, we used the Rietveld method, which is usually used for the correction of XRD measurements in case of any vertical displacement of the samples, which is common because the machine is really sensitive. The statistical analysis was done with a Kruskal-Wallis one-way ANOVA test to check our hypothesis, since the data showed that there was no normality and no homogeneity. To start out with our results, we saw that the highest magnesium mean levels were seen in the Demercinus horridus species, which is part of the subantarctic species, but they had a low number of replicates. The Cetacetarius nutrix came in close second, also a species from the subantarctic, having the highest single value found, which was 10.01 mole percentage. The skeletal component, which had the highest value, was the lantern, so the Aristotle lantern, the mouth, which was also presented the highest mean, and the C nutrix had the highest value mean for the lantern with 9.67 mole percentage. Now, the region that presented the highest levels were the subantarctic, when compared to the Antarctic, with station 80 and Sum having the highest mean values. For the lowest magnesium values, on the other hand, we saw that the Sterecinus neumayeri, which is an Antarctic species, had the lowest mean values, also followed by the lowest single value found in the Notocidaris hemigera, also from the Antarctic. The skeletal component which had the least amount of magnesium content was these spines, with Notocidaris hemigera again with the lowest mean, followed by Notocidaris platyacreta. Again, the region that had the lowest magnesium values was the Antarctic when compared to the subantarctic, with station 72 having the lowest mean of 4.25 mole percentage. When looking at the congeneric species from the different regions, so different species from the same genus, from different regions, we saw that the Sterecinus diadema, which is part of the subantarctic region, had way higher values than the ones from the subantarctic. So they are peers in the Antarctic place. So Sterecinus sp, Sterecinus antarcticus, and Sterecinus neumayeri. The same can be seen in the Abatus genus with a slight difference. Here they are more comparable, but still Abatus cordatus showed higher values, the subantarctic species, rather than Abatus sp species and Abatus cavernosus in the Antarctic. Coming back to our hypothesis, we saw that there was a significant difference between the different skeletal components regarding their magnesium calcite in both levels. So there was difference between the different skeletal parts. However, there was no significant difference between the different species in both regions. This comes to our discussion to interpret these results. So, starting with the skeletal components, we saw that the significant intraspecific difference in the magnesium calcite can indicate that these are more biologically controlled, which was against some of the previous studies done with Antarctic species, as Karate and Garcia produced. Again, Aristotle lantern showed the highest values of magnesium calcite, which could be explained by the fact that high magnesium presence in minerals, in echinotherms body or components are usually related to hardness and robustness of the component. The spines showed the lowest values of magnesium calcite, and this can be explained by the fact that usually they are only covered by a thin layer of epidermis, making them exposed to the surrounding water. If the surrounding water is cold, they tend to have magnesium levels low. But as the seawater starts to rise, they tend to also have their magnesium levels rise. And that's what we can suppose that is happening with the Cetocidaris nutrix, because we could see that the specimens collected at 10 meters depth, so closer to the superficial waters that are technically more warm and acidic, had higher magnesium levels than the ones collected at deeper depths, such as the 599 and 789 meters. As for the test, they showed intermediate values, which is comparable to literature between the 7.4 and 8.9 mole percentage. When we look at the species now, we saw that the highest values again was from the Demercinus horridus and Cetosidaris nutrix. Sadly, we don't have any knowledge about the morphology and physiology of the Demercinus horridus because it was the first study that showed the morphological skeletal components of this species. So we're going to focus on Cetosidaris nutrix. The species have a great environmental distribution range, being found both in low and high salinity environments, also having a high trophic position, which makes it really important in the subantarctic environment. Although they had like the second highest magnesium levels, they seem, and which could put this species to danger, they seem to have a plasticity toward environmental changes such as the pH and temperature. They can switch their feeding habits according to prey availability and also change their habitats or their ecological niches, turning to have a southward direction, so more cold, to more colder places, so in direction of the Antarctic. Similarly, we can see that with Pterechinus diadema, which is the congenital species in the different regions, which was the one that showed the highest values of magnesium calcite being present in the subantarctic when compared to the other species from the same genus in the Antarctic region. It also had an even broader distribution than Cetosidaris nutrix, also having a high trophic position. And they are also able to change their distribution range to more cooler places. The Pterechinus neumayeri showed the highest values of magnesium calcite between the Antarcticus group genus, so comparing them to the Pterechinus antarcticus and Pterechinus species. They also are keystone species in the food web of the Antarctic, but they seem to have a buffer, an internal buffer, that can control the pH level in the body of the echinoderm, which could fight against the ocean acidification stressors. Now for the Abatus cordatus, they don't have the same mechanisms, and they are endemic to the Kerguelen Island, and they don't show high trophic position nor adaptive mechanisms, which could lead to potential extinction in the future. As for the location, we already saw the subantarctic region had higher values when compared to the Antarctic. That can be explained because it's still Eastern Antarctic is still considered resilient. The magnesium content had been associated with elevated sea temperature water, which could be confirmed in the study. Some limitations that we came up with this study was a delay on the shipment of the second set of samples, which pushed back the process of the interpretation of results. And there was also a lack of the same replicates for all the species and a lack of data in literature to be able to compare our results. As for the perspectives, we can see that the next step would be to do a species distribution model, similar to what we've seen in the discussion. And for investigating influences of ocean acidification and marine calcifiers would be to take specimens from past expeditions and new ones to see how the magnesium carries over time. As our conclusions, we have that there were some significant intraspecific difference, Aristotle lantern having the highest values, while Spines showed the lowest. There are significant interspecific difference, but doesn't mean that we can refute this thing completely, just more studies need to be done. We can see that there is a relation between higher magnesium calcite levels and high temperatures, as we saw that subantarctic waters that tend to be more warm have more magnesium calcite, leading to have more prolability to solution. And although some species are able to adapt, others don't, and they are going to be maybe extinct in the future. The take-home message that I wanted to end was that the only way to reduce the climate change and ocean acidification effects on the benthic ecosystem would be to study the physiology and morphology of the study of these species that are still understudied, to try to understand if and how are they going to be able to adapt to the fluctuating conditions in the future. And these are the references, and thank you for your attention.
Thank you. We haven't started yet with you, Nadia, so let's open the floor this one.
I have a question. First, how the temperature is related to the magnesium content?
It's still not completely proved that the magnesium, that the presence of magnesium in echinoderms are related to warmer waters. But some studies show that there is this connection because we can see that in lower latitudes, the echinoderms tend to have higher magnesium calcites than the ones found, for example, in Antarctica and subantarctic regions. There's also even some studies that dare to say that we can use the magnesium as a proxy for seawater temperature, but it's still not really confirmed. With our results, we could say that this association has some basis, but it's not completely scientifically proven yet.
And you said during your introduction, it was important to analyze how these organisms will react to future changes, blah, 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 to help better management of the areas.
Yes.
So, and you also talked about one species that is a good candidate for extinction. So, for instance, what can you do to avoid the extinction of the species that you talked about?
So the Abatus cordatus, which was the species that tend to have more the potential to be extinct, is from the Kerguelen Islands. So now we saw that they have warmer waters than compared to the Antarctic place. And they have a very low niche, ecological niche because they need specific temperatures and salinity. They also don't have a great larval, they don't present a larval dispersing stage, which also reduces their distribution range. So I think that would be important to maybe retract them and put them in another habitat which is cooler, because they are not able to do that change on their own. But this information was based in past SDM models. We would like to do SDM models for our results that we weren't able to do it yet. But I guess that would be the only option, because they are only able to be in the Kerguelen Islands, and if it continues to rise the temperatures, they won't be able to do this change on their own, since they have a brooding type of reproduction. I think I answered.
Yeah. Okay. Paola.
Thank you. This nice presentation. I have two kind of questions. My first question perhaps is more related to this first part. It was the different, why to use the magnesium and why don't use now other ions or cations they precipitate in the test or the other part of the echinoderms in the meantime. What do you think it could be interesting? What kind of information we can have by observing the carbon, for example, or some calcium also that it could be deposits?
So we decided to choose the magnesium first, the first part of the question. The magnesium was chosen because now they have been coming up with more studies with this relation between the magnesium presence and the high temperatures. So my supervisor have done studies in other subantarctic islands, and this one was the first one done on this place with these species that are being first morphologically described in this study, such as the Mercinus ohidus. So that's why we chose the magnesium, but we can also see the calcite or the other parts of the skeleton components, which was going to be done with the XRF analysis, which I told on the limitations. We did the fluorescence analysis, which could give us the whole skeleton components, but we weren't able to assess these results because of the shortage of time in the second set of samples. As you probably know, my study was going to be focused only on subantarctic samples at first. Even the abstract was done only with subantarctic, but then in the middle we decided to add the Antarctic because we thought that could enhance the research. But the shipment of the set of Antarctic species took too long, and sadly we didn't have time to really perform all the studies that we wanted to have a broader results. So,
second question, it's more a link on the species that you can find everywhere with a good shape, a good magnesium precipitation. This species is quite plastic, or it could be very adapted. So how you can manage to know if you have two populations in two sites adapted to two different locations, or is just the same population with a huge plasticity?
We wanted to, we even discussed the possibility of doing a comparison between the first, in the Kerguelen Islands, the two different campaigns were done around 10 years apart. So we wanted to maybe compare the species that were picked in both of these campaigns. But the problem is that the species weren't collected in the same site, so nor the same depth. So the Kerguelen protected campaign, which was the most recent one, was captured at around 8 to 10 meters, while the Phuket campaign done 10 years ago was captured off the coast of Kerguelen Islands at a much deeper depth. So we couldn't really compare that. So I based my results on the SDM models that are already available for the subantarctic species and the Antarctic species. So I don't really know if it's the same population or it's just also suppose is all supposed that they are able to adapt, not necessarily confirmed, because there's still studies being made if they are able to change their ecological niche or not. It would be nicer if we could have the SDM models for our results to see if it goes well with the literature that is already available. But it's already a good baseline for species that haven't had anything yet.
Do you have information on the if like the magnesium content changes with the age of the organisms, from the literature?
The age?
Yeah, like a young organism has the same magnesium content as an old one or not? Does it change with time?
Yes, we saw that usually during the larval stage, the beginning of the life of the echinoderm, they tend to have higher magnesium levels as opposed to when they reach adult life. But we didn't focus much on the phase of life of the echinoderms because the first set of samples from the subantarctic was done with more information. So we had more data about how they did it and how they collected and all of that. While the Antarctic portion was already done and these species were already separated and stored, and the data wasn't really that full to know when or how they developed, right? So we didn't focus much on the magnesium for the life stage in this sense.
I was wondering, you talk a lot about the difference between like the Kerguelen, Antarctic, and the effect of temperature, but you don't show us any data on temperature or other environmental conditions. So do you have those data? What were the temperature range in Kerguelen versus Antarctica?
Yes, I could have added that maybe for a little bit more context. When we were trying to compare the stations, so within the regions, for example, the different stations, we couldn't really have much data about the differences between the stations because the information didn't arrive to us. So we just decided to push that aside between the stations and do more about the regions, the difference about the regions, considering that the Antarctic is colder and the subantarctic is warmer, based on that.
But what are we talking about in temperature? Is it 0 versus 20? Well, what's the difference between the two sides?
Yes. It's they're both known for really low temperatures. Obviously, the Antarctic continent is known for having even lower temperatures, but I can't give you like specific numbers.
Yeah, I think it's always nice when you
Yes.
do a lot of comparison to, you can use satellite data, or there's a lot of database used for that to give a good context.
Okay.
Maybe another one. You talk about the low magnesium calcite in the spines, and you link that to the fact that they are more exposed to changes in temperature.
Yeah.
I'm not sure to understand. Sea urchins don't control their internal temperature, so any part of the body will have the same temperature?
So the literature explains that the further away from seawater, the highest the magnesium value gets. So the spines tend to have the lowest values, while the internal part of the body tend to have the highest. So the Aristotle lantern that is enclosed in the echinoderm's body show the highest values, while the spines show the lowest. And that is because the spines are in direct contact with the cold water. But the cold water getting warmer and more acidic, it tends to influence the magnesium value, making them go higher. Did I answer the question? Not really.
No, I'm not sure to understand, but maybe it's
They don't really have the spines don't really have a protection such as the test, or so they are prone to vary according to the surrounding water in the.
Okay, so it's more chemically driven rather than temperature driven.
Well, the chemical is dependent on the temperature, right?
Well, I'm thinking more about the chemistry of the water, like pH and that would impact more
that's why
the parts that are exposed than the internal parts where there's a better control of what's happening.
Yes.
But the temperature itself of the organisms, the temperature at which precipitation occurs, is the same for the spine or the internal part of the body, since they don't control the internal temperature, right?
Yes, but the spines are directly in contact with the water, while the internal part not. That's why we think that it's more mainly biologically induced than environmentally induced
Okay. Yep.
for the internal part. While the spines, we suppose that it's because of the higher temperature that is going to influence the spines, and it was proven in literature. It's comparable to previous studies that already show that the spines are starting to have higher magnesium values than before. But it's more, we said that it was mainly biologically induced because it was more about the robustness or the hardness of the component rather than the environment itself in the skin.
We want to conclude.
For sure. Thank you for the very nice presentation. I'm not sure I have a specific question, but just if I understood well at the beginning of the presentation, the samples that you had some were from 2008, some 2013.
Yeah.
So, and that was one of my questions about the temperature. It's almost impossible to compare these samples because I don't know at all about the temperature of the ocean back in even now or back in.
The first set of samples we were going to analyze were just from subantarctic Kerguelen Islands Protector campaign, which was done 2022-2023. But the process of dissecting and doing everything was pretty fast and ended at around March. So we had still a lot of time for the internship. That's why we decided to add the Antarctic species that were available from these past campaigns, but they weren't studied yet. So it was interesting because the poker was from the 2010, which would be interesting to compare, like what I told Paula about how the magnesium changed, but one was near the coast, the other was off the coast. It was really difficult because they weren't done with the same, methods, or in the same regions even, or the same regions or the same locations. So it was difficult to compare in that sense, but yeah,
You had no choice.
Yeah.
For that, so
yeah.
and no, I think it's okay. The question was relevant. Thank you.
Okay, so we can jump back to previous to remember if you want, for a couple of questions. I trust Paula has.
Yeah, sure. Well, more technically, could you go back to just one experiment when you show the link with the species, with the area, this one, for example, this one. How did you test it exactly? And did you try to do some clustering with your data, or you test the different categories by themselves?
We tried to do a PCA, which we discussed, but the stations were just giving different results, and we just decided to take the stations out of the question because they weren't really in the same category. As I said, the methods weren't the same, so we just decided that it was going to be too biased to have preciser results. So we just focus on finding the highest, the lowest values, doing the box plots, and trying to see from these three aspects what we could find from our results.
If you go back to the pictures when you see the variants within the same genus, for example, this one, you say that you have higher magnesium content, and I didn't catch in which species and how you test it.
Okay, so we decided these are the only two species that we could find in both regions. So both in the genus that we could find in both regions, both in the Antarctic and the subantarctic. This Pterochirus diadema was part of the subantarctic region, the only one. And we could see that they had higher mean values, both for TESS, Aristotle lantern, and spines. Here is not separated by skeletal component. It was just all the whole mean of magnesium values found.
Mean, and how you tested that this mean is higher?
So yes, the magnesium content can be done by LMC, which is low magnesium content, IMC, intermediate magnesium content, and HMC, which is high magnesium content. So we saw that the subantarctic species, while with also the lantern, showed the HMC range, so it's above 7 moles percentage. while the intermediate was between 4 to 6, and the low was 4 to 1. So that's how we decided which were the highest, the intermediate, the lowest, and this is how we also described in the discussion here. So basically, high HMC, LMC, and
I can really test this different distribution in the data.
Yeah. And just, it could have endless possibilities of what to do, but we just had to pick things to focus on, but obviously there are a lot of other things.
Okay, thank you. Is this better? So at the end of this first part, before you leave, just a few information. This afternoon, we'll go for the second session, which is ecosystem and biodiversity. We'll start with Cassandre, who is online. The session is called, so not online. So it means if you're on site, you can participate. You can, it will not be online. So for the people following online, we will join this afternoon with Isaac at 2:10. But of course, you're welcome from 1:30. So this afternoon will be with Cassandre, Isaac, Roman, and Emma. Cassandre and Emma will be online because they are far, far away. And so we will be here in the salle des actes. And for the lunch, as the weather is not really Côte d'Azur today again, Vincent preferred to set everything with the guys in the room just there. But of course, you can have a look around to get fresh air. Okay, so we are back here at before 1:30 because the next presentation is at 1:30. Bon appétit, à tout de suite.